with chapter 5, we're going to talk about metabolism. And remember, when we talk about microbial metabolism, this covers bacteria, which are prokaryotes, but it also covers fungi and protozoa, and those are eukaryotic. So we have to cover both types. Also, among bacteria, we have different types, some that live in oxygen-rich environments and some that live in the absence of oxygen and some that can do both. So we have to come at it, we have to have a complete picture of these different types of metabolism. And then, of course, we also have different types of food, right? We have our carbohydrates, our protein, and our lipids. So it is kind of a heavy subject, but I've tried to simplify it as much as possible, okay? Before we can talk about metabolism, we need to have an understanding of enzymes, so we're going to start with enzymes. All right, so collision theory states that in order for chemical reactions to occur, atoms or molecules must collide, and they must do so with enough energy and the correct orientation to cause a rearrangement of stable electrons, right? So you're taking two atoms or two molecules that are already stable and you want them to react with each other, and so you have to force that, and you do that with enough energy and the correct orientation. And so usually this means that lots and lots and lots of collisions have to occur before the correct one will actually happen and cause the reaction you want. So if we wanted to speed up the rate of chemical reactions, like in the laboratory, we would do things like increase the concentration of the reactants, increase the temperature or the pressure, and then that would force more collisions to happen and increase your reaction rate. But in the body, we cannot do that, right? We have to keep a somewhat constant body temperature. Our body temperature can fluctuate a little, but we couldn't just keep increasing it more and more. So we need enzymes, and we'll talk about that a little bit more in a second. Okay, so activation energy is the amount of energy that you have to put into a system in order to cause the chemical reaction to happen. And remember, a chemical reaction, when you're talking about regular atoms or regular molecules, these are already stable. The electron configurations are complete, stable, right? And so if you want them to react with each other, you have to disrupt or rearrange those configurations, and so that takes energy. And so I already talked about reaction rate, but it's the frequency of collisions with the right amount of energy and the right orientation to bring about that chemical reaction. And so, as I mentioned, if you want to increase the reaction rate, you could increase temperature, pressure, or the concentration of the reactants, which are great if you're in the lab, but in the human body, we can't really do that. So we use enzymes, right? And so the activation energy is the amount of energy that you have to add to the system to get that chemical reaction to happen. And it's a very high amount of activation energy to take. In this situation, you're taking one big molecule and splitting it into two separate molecules. And you can see in red with the enzyme, you're able to carry out that chemical reaction without adding as much energy. And in this diagram, it's half the amount of energy. So why is it that enzymes can lower the activation energy so much? Well, it's getting the orientation perfectly. So that substrate, the reactant, is going to go into what we call the active site of the enzyme, and it will only fit in that active site in one exact position. And so by having it in that perfect position, you're able to carry out that chemical reaction in this case is also splitting it into two separate products. And again, because the orientation is perfect, and for other reasons that we won't discuss in this class, it lowers the activation energy. So when you take biochemistry, if you are getting a 4-year degree, you will probably take biochemistry, and then you'll add on to what you're learning now. Okay, so the definition of an enzyme is that it's a biological catalyst. Catalyst is something that, speeds up a reaction. And so biological catalysts are enzymes. Enzymes are protein, but they can also be a protein with a inorganic compound attached to it. Enzymes are highly, highly specific. So this active site is only going to fit this substrate. You could throw lots of different substrates at it and nothing will happen because it's specific. It's like a lock and key. And so once the reaction happens, it's free to keep going over and over again. So not only are they fast, they lower the amount of energy, but they can keep, enzymes can keep carrying out that reaction over and over again until the substrate is gone. Okay, what would slow down an enzyme is if you had very little substrate and it took more time before the substrate and enzyme come into contact. So I mentioned that a lot of enzymes have a non-protein portion, and that would be called a cofactor. So some enzymes, few, are just protein and there's nothing else to it. It's very simple. And then that would be a functional enzyme. But most enzymes actually have an additional component called a cofactor that's required. And so unless it has the cofactor, in this picture, it's a coenzyme. And once that cofactor is bound to the enzyme, it creates the rest of that active site. And now this entire unit, the apoenzyme and the coenzyme, are referred to as the holoenzyme, as in whole, right? And now it's active. So this picture shows you the apoenzyme, which is just protein, but it is not active, it's inactive. And then a vitamin, an organic molecule called a coenzyme, comes in, binds to the enzyme and makes it a whole complete enzyme that is now active. And you can see it completed that active site. Sometimes they're not actually part of the active site. They might bind somewhere else on the enzyme, which causes a change in the shape of the enzyme, which then is how the active form comes to be. Sometimes these cofactors, instead of being a vitamin, they're just a simple metal or ion, right? So it could be iron, zinc, magnesium, which are all metals, or calcium. Okay. So let's think about the function of the enzyme, right? So the very first step, once you have your active enzyme, is for the substrate to bind to the active site. Then the enzyme substrate complex is now formed, and now you can get this change in the arrangement of electrons in the substrate, and that's going to then lead to the release of your product. Okay, so as seen in this picture, actually the picture is kind of back here. You have your active enzyme, the substrate enzyme complex is formed which will lead to a change in the arrangement of electrons in the substrate. So that's the actual chemical reaction happens, right? And as soon as that chemical reaction happens, then it's going to release the products and it's going to be free to do it all over again. So the enzyme is not used up. It's free to repeat this over and over again. Okay, so already mentioned that enzymes are very fast. A super slow enzyme would be one reaction per second, but typically our enzymes are closer to 10,000 reactions per second. Okay, so only the specific substrate can fit into the active site of the enzyme. The enzymes are specific based on that shape. And so because the function of the enzyme is based on shape, that should answer this question. Adding heat can speed up a reaction, but too much heat will break the hydrogen bonds in the protein part of the enzyme, and that causes the enzyme to denature, and then it'll no longer be functional because it has lost its shape, right? So if you break the hydrogen bonds in this protein portion of the enzyme, it'll look something like this: unraveled, unfolded. So why is it that we can add a little heat to speed it up, and we have to be careful not to add too much heat, right? But if you add just a little heat, then everything's moving faster and the substrate is going to come into contact with the enzyme more quickly, and so that can increase your rate, your turnover rate. Okay, and so that's what we see here. As we increase the temperature, substrates and enzymes are moving faster and they come into contact more often, and as soon as they come into contact, the reaction will happen. And so you'll notice that the optimal temperature, the fastest that the enzyme performs, is getting pretty close to the cutoff of denaturing. And so you have a steep drop off when it gets too hot because you start to break those bonds. Now, why isn't this an immediate straight line? Because we're not talking about just one single enzyme. When we're talking about things in the body, things in a cell, right? Even just one cell is going to have enough enzymes that you won't denature every single enzyme instantaneously, right? But they will start to denature, and that's why you get this steep drop off. Okay, pH is a little different. It's a little more even on both sides. So becoming too acidic or too alkaline can denature your protein, right? Because it's also going to change the interaction of bonds. In this case, now we're thinking about the amino acid side groups and how they behave with one another. And this example is showing an enzyme with an optimal pH of 5. But remember, different parts of the body or different cells that live in different environments, right? They're going to have different optimal pH levels. Okay. Same with this example. So we're thinking about the human body. We see an optimal pH, optimal temperature, of 37 degrees Celsius, which is body temperature. But if we were to look at a bacteria that normally lives in the soil, its optimal temperature would be lower, be closer to something like room temperature or even cooler, right? Okay, so you always have to think about the situation in which the organism lives or the enzyme functions. If we're talking about a person, which compartment of the body are you talking about? If we're talking about a bacteria, is this a bacteria that grows on us, on our skin? It will be cooler. then internally inside the body, right, will be warmer. And of course, we are much warmer than like the environment, soil, water, etc. Okay. This last graph that I want to show you regarding enzymes shows that as you increase the concentration of your substrate, you will increase the turnover rate, right? The reaction rate of that substrate and that enzyme, right? So as substrate concentration increases, your enzyme activity will increase up to a certain point where then it remains exactly the same. No matter how much more substrate you add, it's not changing. And so this point right here where it becomes the same enzymatic rate is called the saturation point. It means that all of the enzymes are bound with substrate, so adding more substrate is not going to make a difference. What happened between this point and this point, when we did see an increase in enzymatic activity, that meant that by adding more substrate, you were increasing the likelihood of a substrate and an enzyme to come into contact. Or in other words, decreasing the amount of time it takes for enzyme to reach the substrate, right? And then you reach a saturation point, and it doesn't matter how much more substrate you add, all of the enzyme is busy, okay? So please do not confuse this with a common mistake is that people see a straight line and they think nothing's happening. On the contrary, an amazing amount of things are happening, right? Every single enzyme is working to release product and repeat over and over again as fast as it can. Okay? So now that we've learned the basics of how enzymes function, we can start thinking about ways in which that function might be inhibited or slowed down or stopped. Okay? So normal binding, as we just talked about, is when the substrate binds to the active site. If you have a competitive inhibitor in the solution or environment that this enzyme is located, the competitive inhibitor can bind to the enzyme, and then that's going to prevent the substrate from being able to bind. So see how this competitive inhibitor is taking up part of the active site? It's not taking up the entire active site because it is not the actual substrate, but it has a region that's identical that's able to interact with this region of the active site. And now the substrate is being physically blocked from binding to the active site. Okay, here's an animation that shows you this concept. So we see this happens a lot in defense mechanisms to basically kill off bacteria, fight with other bacteria to slow down or kill off, so that they can get the resources and take up space, right? We also use these strategies when trying to slow down or kill bacteria ourselves when we have an infection, right? So PABA is right here, okay, and it's used to make folic acid. Folic acid is a coenzyme for many enzymes, okay, and it's especially important to be able to make DNA or repair DNA that's already made. And in animals, it's also vital for red blood cells And so this is one reason why, for instance, when women become pregnant, or if they are trying to become pregnant, they would go on prenatal vitamins. And one of the main ingredients is folic acid. Okay. So notice that this molecule on the left looks very similar, right? It has the same NH2 aromatic ring, and then it's just a little different on top. And so this sulfonamide is an antibiotic drug that mimics PABA, and so it will kill bacteria by being a competitive inhibitor. Okay, so other types of inhibition would be a non-competitive inhibitor, where the binding is now somewhere other than the active site. So they're not competing to who gets to the active site first. The non-competitor is binding to somewhere other than the active site. And in either of these cases, competitive and non-competitive, the amount of time that the inhibitor stays bound to the enzyme can vary. So that's another thing that you need to take into consideration is it permanently bound, or is it constantly binding and releasing? Feedback inhibition is a common, I don't want to say pathway, it's a common mechanism. There we go. Feedback inhibition is a common mechanism used throughout different metabolic pathways and all sorts of functions in the body. And you're already familiar with feedback inhibition in a non-biology way, right? When you use central air or central heat, right, you have a thermostat, and the thermostat is constantly measuring the temperature of the air, and if it gets a degree above or below whatever you've set it to, it will kick on, right? So if I'm running my heater at 70 degrees Fahrenheit and it becomes 69 degrees in the house, then it will kick on, and it will stay on until it reaches 71 degrees, and then it will kick off, right? And it constantly does this back and forth. And so it's a feedback inhibition in that it only turns off once you have enough, right? So here you have a substrate, it binds to an enzyme, and then it goes to another enzyme, so that you get a first enzyme and some product, and then that goes to another enzyme and then another product, and then another enzyme, and you get your final product. And this will keep happening over and over and over again as long as there's substrate. But what a negative feedback inhibition says is that if the end product starts to build up, you start to have more of it than you need, it starts to just sit around, it can act as an inhibitor to the pathway, the very pathway that's producing it. And so you have a bunch of the end product building up. It starts to bind to the what we call the allosteric site, and that's going to block this enzyme, right? It changes its shape, and now the substrate no longer fits, and that pathway is shut down, but only temporarily. Once you use up this product, there's less of it to bind to enzyme, and so now more enzyme will be free to do the work. And so when we say shut down, it's not 100% off. It's going to slow it way down, and until it's needed, and then it will speed up. And so it's constantly going back and forth, back and forth. And this gives you much more fine control rather than actually flipping a switch and turning something on, flipping the switch again to turn it completely off. This is more on a scale of making a lot, making less and less, more and more, less, more, right? Depending on how much of the end product is binding to enzyme. And again, you have to remember, we're not talking about just one individual enzyme, even though that's what's shown in your images, but there would be millions, right? When we talk about molecules, we use one in an image as an example, but you would never have just one in an actual situation. Okay, so factors influencing enzyme activity. If the temperature gets too hot, it'll result in, that's right, denaturization. If it gets too cold, what happens? It'll just slow down, and it gets slower and slower, and if it gets to freezing, then it will actually stop. But you're not ruining the enzyme. You can always warm it back up and speed it up, right? So too cold is fine. It's not going to damage the enzyme. But if you get too hot and you break those hydrogen bonds, it's denatured and it's gone forever. Okay? Number 3, optimal temperature will do what to the reaction rate? So the optimal temperature will increase the reaction rate, and it will do this to the maximum. It'll give you the maximum reaction rate of the enzyme. Okay? And that will be different for each enzyme. An increase in the substrate concentration will increase the reaction rate until you reach the point of saturation. Optimal pH will also increase the reaction rate. And of course, if a cofactor or coenzyme is required, then increasing the amount of that will also increase your reaction rate. And if you have a competitive inhibitor, you're going to slow down the reaction. So the presence of an inhibitor will slow down the reaction. And it could be permanent, or it could constantly bind, release. Okay? If it's a permanent binding, then that enzyme is now no good, and you would have to make more enzyme to replace it. All right, enzyme classification. So I'm not going to test you on this, but you might see these throughout the course and in other courses. So an oxidoreductase is a oxidation reduction reaction. It's the category of oxidation reduction reactions. Common ones are like dehydrogenase. You may have already heard of dehydrogenase in other classes. That's how common it is. We have things like transferases, where you're adding or removing a kinase, or you remove an amine group, so a deaminase. A kinase can add or remove a phosphate group, depending on the situation. Hydrolase, like hydrolyzing something, okay? These are usually the specific substrate that it works on, or the product that it makes will appear in the name. But hydrolase is going to be a hydrolysis reaction. Lyase, removal of atoms without hydrolysis. We see this with like decarboxylase, which we'll see later today. Remember that lyase, so decarboxylase, so this has a little typo. This should be YLASE. Right? So it ends with olase. When you're removing atoms without hydrolysis, it's going to be a lyase, as in decarboxylase. Isomerase, rearrangement of atoms. And I don't know that we'll actually deal with isomerase in our class, but when you study DNA synthesis, so like if you take 4A, for example, you would see this. And ligase, same. We will actually see ligase in this class. And so this is ligating something together, joining it together. Okay, this requires ATP. We also have acetyl-CoAse synthetase and DNA ligase are examples of ligases. And DNA ligase is something we'll see later in this class. Okay, so again, I'm not asking you to memorize the classifications of enzymes. I'm just showing them to you so that you become familiar. So when they do come up, you'll be somewhat comfortable with them. Okay. Now, this you do need to memorize. Important coenzymes in cellular metabolism. You're going to see this today a lot in this chapter. So NAD plus comes in and then takes some electrons and becomes NADH, right? NADP plus, also same idea. We'll add a hydrogen to it. And FAD, this really should say FAD plus, and then it will become FADH2, so it can take up 2 hydrogens. And then coenzyme A, which a lot of times we just will abbreviate CoA. CoA plays an important role in synthesis and breakdown of fats, but it also plays a key role in being able to utilize the Krebs cycle in carbohydrate catabolism as well, as you'll see. FAD, you might be familiar with. It's a derivative of one of the B vitamins, riboflavin, right? NAD plus and NADP are derivatives of the B vitamin niacin. So this is why you may have heard that taking B vitamins can increase energy. So some people will take B vitamins to give themselves a little energy boost. And that's because the idea is that you can increase your rate of cellular respiration, make more cell energy, and that will in turn make you feel more energetic. If you already have plenty of the B vitamins in your diet, then you might not actually have that effect. But most people don't eat enough fruits and vegetables every single day, and so a supplement every once in a while might actually be able to provide that extra energy to you. Okay, so catabolic and anabolic reactions. So remember that I said in order for metabolism to work, we need enzymes, and then that was kind of all I said about it, and we've talked about enzymes this whole time. So now we're moving into looking at metabolism. And metabolism is the set of all of your chemical reactions within a cell. And then when we talk about people, then we're now adding all the cells of all of the tissues and all the organs all together for your whole body to function as a whole. Okay. So we break these into catabolic reactions, which release energy, and anabolic reactions, which take energy. And because you have one that's producing energy and one that's requiring energy, we see that metabolic pathways are coupled. As in, what that means is that they're happening in the same place at the same time, side by side, so that as energy is being made and released, it can immediately be utilized. So if you don't need energy, you will slow down the production of making energy, which is why you already know this concept. If you eat more calories than you actually need, you will store them up, right? First, we store up glycogen, but our body actually can only hold a small amount of glycogen because the amount of water that it attracts. And so then all the rest of it goes to fat, right? And this is why when you want to lose weight, you're told you either need to eat less calories or you need to exercise more to burn more calories, or you do both, right? So yeah, all of these principles that we're learning can be applied to us as humans, as well as thinking about single-celled organisms like bacteria. Okay, so I think this is actually a good stopping point before we get started with more about metabolism. So go ahead and take a break and then review what we've already done so far, and then watch the next portion when you're ready for more. All right, good job.